Hello students, in today's session we'll go through certain concepts in toxicology. Toxicology. It is a branch of pharmacology that deals with the study of adverse effect of substances or chemicals on living organisms and assess the probability of its occurrence. Toxicants are the naturally occurring or synthetic xenobiotics that are capable of giving toxic responses. Toxins are substances of biological origin that elicit toxic responses. Poisons are toxic substances, but they are inorganic in nature, such as lead and arsenic. Paracelsus is known to be the father of modern toxicology, and according to him, all substances are poisons. There is none which is not a poison. The right dose differentiates a poison and a remedy. Toxicokinetics is a subgroup of toxicology that deals with what a body does to a drug when given at a relatively high therapeutical dose.
The field of toxicology is further divided into many subfields based on the area of applicability. Example, clinical toxicology. It deals with diagnosis and treatment of diseases and effects of xenobiotics on organisms. Occupational toxicology, which deals with chemicals an individual is exposed to at his or her workplaces. Example, the packaging industry and the paint industry. Environmental toxicology, which deals with the effect of pollutants on, on living organisms present in the ecosystem. Product development toxicology. It deals with the toxicity studies and strategies taken for production of a product from the product discovery to its commercialization. It includes studies like ADME, general toxicity, genetic toxicity, immunotoxicity, reproductive toxicity, bioassays, and microbiology to ensure human safety. A toxic substance can be a systemic toxicant or an organ toxicant. It depends upon its site of action. A systemic toxicant affects the entire body. Example, potassium cyanide. While a organ toxicant affects only target organs or specific tissues. Example, benzene, primarily toxic to blood-forming tissues. Lead to three target organs, the central nervous system, kidney, and the hemopoietic system. Toxicology studies are based on dose-response relationship. Higher the dose, more severe the response. The magnitude of toxic response is therefore proportional to the concentration or amount of the chemical at the target site. The four important processes that control the amount of chemical that reaches the target site are ADME. Toxicity of a chemical depends upon its dose. It could be a measurement of environmental exposures or the amount of the chemical or substance administered over a period of time. There are three types of doses, that is the absorbed dose, the administered dose, as with the total dose, which is the sum total of all the individual doses that are given. The degree and the spectra of response depends upon the dosage and the organism. A response can be local or systemic, reversible or irreversible, immediate or delayed, graded or continental. It could be at the molecular, cellular, organ or organism level. A general toxicology study undertaken as per the OECD guidelines are given below as a flowchart. The test substance is administered to the animals and these are maintained under lab conditions. The experiment needs to be designed based on the dose level, dose selection, and type of route of administration. The parameters that are measured during this period are the body weight, food intake, water intake, clinical observations, clinical pathological observations, biochemical studies, and gross necroscopy. The data analysis is taken and statistically the data is reported. The common clinical observations taken and recorded are given below. They are, to name some, respiratory, motor, convulsions, reflexes, skin, cardiovascular signs, etc. The different type of toxicity studies are single dose or multiple dose studies, which includes acute toxicity studies. Second, repeated dose studies, which include subacute and chronic toxicity studies. Next, local toxicity studies, allergic or hypersensitivity toxicology studies, genotoxicity studies, carcinogenicity, and reproduction toxicity. In these, we'll concentrate only on the first two types of studies, that is, acute toxicity studies, subacute and chronic toxicology studies. In toxicity studies, the acute, subacute, subchronic or chronic studies depends upon the duration of exposure of the organism to the particular test substance. Example, for acute 14 days, subacute 28 days, subchronic 3 months and chronic 6 months. Now, these chemical substances or test materials are given orally by inhalation, dermal or other methods as prescribed for administration into the human body. In the toxicity studies, the endpoints or the outcomes that are recorded are due to these exposures, the effect on or response on the target organs, the biochemical functions that get altered compared to the normal, the physiological functions that get altered compared to the normal, metabolites generated, specialized acute tests for irritation and sensitization, then certain effects of the test or exposure on the reproductive capability, the genotoxicity test. For all the toxicity studies, one of these would definitely be a endpoint or outcome. Acute toxicity studies. The objective of these studies is to identify a dose that causes adverse effects on the organism and estimation of a minimum dose that causes lethality. It can be divided into two, preliminary and definitive. Acute toxicity preliminary studies. Acute toxicity studies are conducted in early stages of preclinical trials. It is carried out on a rodent, usually a mice or a rat, and on a non-rodent, usually a rabbit. Single or more doses of the test material is administered for a period of 24 hours and the effect is monitored for 14 days. The doses are calculated based upon the body weight. Mortalities are recorded and the morphological, biochemical, pathological and histological parameters are investigated and compared to the normal or control group. The LD50 value of the test substance is also estimated.
An important toxicity index that is measured by acute toxicity study is the LD50 value. That is, the concentration that causes the death of 50% of the animal population that is given a particular dose of the test substance. Various methods are utilized to calculate the LD50 value, but the most common one is the graphical method as shown in the figure and the Carber's method. The limitation of LD50 is that a wide variation is seen among species. Certain test materials that may show toxicity in a rodent may not show toxicity in a non-rodent or vice versa. Example, chocolate, which is non-toxic to humans, are toxic to certain animals. The preliminary acute toxicity test also helps to determine the maximum non-lethal dose. It is the highest single dose which does not induce the death of an organism. In the recent years, the Organization for Economic Cooperation and Development, OECD, has modified certain methods like the fixed dose procedure, the acute toxic category method, and the up and down method. As per the OECD guidelines, the endpoint of the experiment may not be death, but an evident toxicity. Acute toxicity study definitive procedure. In this study, a group of 20 animals of both the sexes are selected and dosed at maximum non-lethal dose. After 48 hours of exposure, 5 individuals of both the sexes are dissected to study the early pathological stages. The remaining animals are kept under observation for 14 days and parameters like change in body weight, food consumption, symptoms of toxicity, etc. are noted. These are then dissected after 14 days. By this method, the target organs can be identified and the maximum tolerant dose and the medium lethal dose is calculated. The maximum tolerant dose or MTD is the highest dose of the test substance that can be administered to the organism without leading to mortality or lethality due to toxicity. The medium lethal dose is the dose that causes 50% of death or mortality of the exposed population to the test substance. I had discussed about this while I talked about the Contal dose response curve in my earlier lecture. The acute toxicity definitive study helps to calculate the therapeutic index, which is the ratio of maximum tolerant dose to the minimum curative dose. Smaller the ratio, better the drug. Repeated dose studies for toxicity. Repeated dose studies are done for subacute and chronic toxicity studies. As per the OECD guidelines, short-term repeated dose toxicity should be done. For both subacute and chronic toxicity studies, these parameters and guidelines are followed. First, the experimental animal population should include 2 mammals, out of which one should be a non-rodent. The duration of the study varies from 30 days to 180 days. For subacute toxicity study, the duration is 28 days, while for chronic toxicity study, it is more than 90 days. The dosages are selected based on ED50 and LD50 values and are categorized as high dose, low dose, and medium dose along with the control dose group. The drug is administered through clinical route and individuals are monitored for their behavioral, physiological, biochemical, and histopathological parameters. The data is recorded. The repeated dose study is used to determine the maximum tolerant dose and the nature of toxicity. The figure here shows the flowchart for studying subacute toxicity. The study group is selected, which include mammals, one of which should be a non-rodent. They are grouped based on dosage given as control, low dose, medium dose, and high dose. It includes 5 each of male and female individuals. The study group is monitored for its behavioral, physiological, and biochemical parameters. After 28 days, the individuals are dissected and the histopathology studies and data is recorded. Conclusions are based on these findings. In chronic toxicity studies, the test compound is administered for over more than 90 days and the animals are observed periodically. The chronic toxicology studies provides inferences on long-term effects of the test substance on animals and based on that, conclusions of human safety can be determined. The chronic toxicity study can be used to determine toxicological indices like the maximum tolerant dose, highest dose tolerated, no observed adverse effect or level dose, and lowest observed adverse effect level. The MTD value is the highest dose that can be tolerated without significant lethality. The HTD is the highest dose that can be expected to yield result relevant to humans. The graph here shows the NOAEL and LOAEL values. Chart here summarizes the different types of short-term toxicity and long-term toxicity studies, the durations of administration of the test substance for each, and the inferences that these studies give. Certain terms that are used in toxicity studies are toxicokinetics. It deals with absorption, distribution, biotransformation, and excretion of chemicals, basically ADME. Toxicodynamics. It deals with the biochemical and physiological effects of chemicals to the body and its mechanism of action. Basically, what the drug does to the body. Bioaccumulation. It refers to the accumulation of chemicals or toxins within the tissue of an organism. Biomagnification refers to accumulation of toxins in organisms in food chain and its magnification of hundreds and thousands of times as it moves up the food chain. Thank you.